Welcome to this week's Lunch and Learn. Delighted to be welcoming back one of our past alumni and a researcher that we are funding both in 2024 and 2025, Dr. Arutha Kulasinghe. Before I talk about Arutha and kick off this session, if you do have any questions, please drop them into the chat box. You can see a chat icon at the bottom there, and I will weave those into the conversation we have with Arutha. The session is going to take about 45 minutes as well. It will be recorded, so if you need to revisit it or you've forgotten anything from it, we'll be sharing that recording with you early next week as well. So, as I was saying, we're delighted to have Arutha here. He is a group leader at the University of Queensland and the scientific director of the Queensland Spatial Biology Centre at the Wesley Research Institute. Arutha has led pioneering studies on particularly lung, head and neck, and now skin cancer using cutting-edge spatial mapping technology. He believes that immunotherapies, which stimulate the immune system to fight tumours, are a game changer in cancer treatment. And I'm delighted to speak to him today about how this happens. So before we start on the scientific stuff, first of all, welcome, Arutha. Great to have you here today. Could you tell the audience, perhaps there's some here that aren't familiar with you, why did you get into cancer research in the first place?
Lovely to be here, Nikki, and you know, we often catch up. It's a great forum that you have here. Just maybe first of all, Nikki, I wanted to just celebrate you and congratulate you on you know your 6 years at Cure Cancer and all the wonderful work you've done. I think you know, we often get reminded of you know, you should give flowers to people that you know often work with, and I think you know, you're one of the most transformative CEOs we've worked with in this sort of early career funding space, and the work that Cure Cancer does is integral for everything we do in the lab. So I just wanted to celebrate you first of all.
Oh, thank you. Thank you. It's a privilege and a pleasure to be able to work along a great team I have here at Cure Cancer and to be able to fund such innovation as yourself. So thank you very much for those kind words.
Absolutely. So Nikki, so we got into, well, I think I was around 10 or 12 years old, and then my granddad had colorectal cancer, and so I wanted to be part of that, you know, journey. I wanted to know what was going on, and so I went and set out surgery. It was in Sri Lanka at 3 a.m. and the surgeon came out with the silver bowl, and he had my granddad's tumor in the bowl, and it was the size of a football. And so at that moment, you know, I just kept recalling that, and it was something that really shaped everything I did from that point on. You know, why do these tumors grow to that size? You know, why they found in those regions? Was it something to do with their diet? All these sorts of questions when you're really impressionable at that age. And so I realized I was growing up in South Africa at that time, and in South Africa, the two biggest, well, in Africa, the two biggest killers are HIV and malaria. But I really wanted to get into cancer research and understand what, you know, the complexities around cancer. And so I did my PhD in Australia in the liquid biopsy space. And so what that means is we look at a patient's blood sample and we try to look at biomarkers within the blood that could be proxies of disease. And so can we track mutations and things like that in the blood? And you know, when we see that certain mutations are increasing over time, those patients might do poorly on therapy, or they might be early predictors in that space. And so that was where my PhD was done. And then I realized really early on that we needed to go back to the patient's tumor and really understand every cell within the tumors. We often, you know, when cancer researchers and clinicians and all of us think about cancer, when we can't explain a concept, we say that it's the heterogeneity of the disease, which means that there's a lot of stuff going on. We don't really understand it. And so I fundamentally wanted to figure out, can we characterize these tumors cell by cell? And so it was a right question at the right time, I guess, where all these new technologies were coming into this space and this field of spatial biology was evolving. And so that's where I set up my lab. And Cure Cancer was fundamental to that first year of funding, because it really let us build the lab and the resources around it. And so what the lab does, Nikki, is we map patients' tumors cell by cell. And so if you look at a patient's biopsy sample or a surgical resection, we're looking at on a section of tissue, we're looking at about 3 to 5 million individual cells that have different locations. And so what we try to do in the lab is to identify what is that cell, what is each individual cell, how does it communicate with its neighbor, and then what is the composition of that patient's tumor, and you know, are there areas that might be sensitive to drug? Are there areas that might be resistant to drug? And can we characterize that from what's used clinically?
And we talk about this and the ability now to see cells in such great detail, but could you just wind back a bit to talk about what treatment was like, or is in some cases still like, for patients? So it's not as detailed as we're doing now, is it?
No. So, I mean, traditionally, you'd be given, you know, depending on the volume of disease, you'd be given surgery, chemotherapy, radiation therapy, and so on. Those are sort of your traditional mainstay therapies that are being given. But the survival rates haven't, you know, significantly improved over the last period. And so for lung cancer, for example, the 5-year survival is still 15%. And so, this really was transformed in 2017 with the work developed by Jim Allison and others, showing that these immunotherapies, which use or harness your body's own immune cells to kill the cancer. So they essentially decloak cancer cells. So cancer cells love hiding. And so what these immunotherapies do is they decloak the cancer cells, and they're able to then be identified by the body's immune cells to then go and kill those tumor cells. And so what we see is that immunotherapies have led to, you know, words such as cure being used for the first time in cancer and in cancer therapies. And so in certain patients in lung, head and neck, bladder, melanoma, non-small cell lung cancer, even some triple negative breast cancers, we're seeing that about, you know, 15 to 20% of patients have long-term benefit. And so what we're trying to figure out is, what is it about their tumor biology that's making it sensitive to these drugs? And so immunotherapies are sort of this new wave of therapeutics that's really forging its way forward, but it's leading to exceptional responses in patients. And, you know, you're not giving a foreign agent as with chemotherapies, you're actually harnessing your body's own immune cells to kill the cancer and identify that as you know, as foreign, and then destroy that by your body's own immune cells. And so immune checkpoint inhibitors is what these are called. They typically cost $300,000 to $400,000 per patient per year. And so, you know, if they only work in 15 to 20% of patients, it's really important to identify those 15 to 20% and then spare, you know, the patients that might not require those, and identify alternative therapies for those patients. So you've got these new therapies that are evolving rapidly. They're very personalized based on your patient's individual tumor, and then we try to identify ways in which we can identify the correct patient for the correct immunotherapy.
Okay, lovely. Thank you for explaining that. Traditionally, would it be fair to say that most patients had very similar treatments prior? And now this, as you say, gives us the ability not only to analyze every individual's tumours differently, but then also personalize that treatment, whether it is just chemo, a mixture of chemo and immunotherapy, or immunotherapy by itself as well.
Yeah. So immunotherapies generally, Nikki, rely on your immune cells to infiltrate the tumor. So the tumor cells should be recognized by the immune cells. And when we see that on the tissue, we know that those patients will do well on these immunotherapies. So it's really identifying the right therapy for the right patient, and really, rather than giving this sort of blanket approach of every patient gets this drug, rather tailoring it to their unique disease and the unique biology of their disease. And we think, and we can see that this personalized approach of personalized medicine or precision medicine is probably going to treat your cancer more individually and target your unique tumor specifically. And that's likely to lead to longer term outcomes.
Absolutely. So can we talk through a question here about the process? So if there is a patient there that has a tumor, where do they go from that, from seeing their clinician, and how does it get to you? And then how do you do that analysis?
Yep. So at this stage, Nikki, most of our work is research use only, but we're building the evidence base for that. For the past 10 years, the real focus has been on, sequencing tumors and really understanding the genetic makeup of your cancer. And so that has given us new insights into the genetic basis of the tumor. But what we're realizing is that, so what my lab really does is we profile the same tissues using what's called spatial mapping. And so spatial mapping localizes every cell in the patient's tumor. So we not only know the genetic makeup of the tumor and each individual tumor cell, but we know where they are, how they're talking to their neighbors, the environment which they're in, the degree of immune recognition of the tumor. And we think that this additional layer of information is going to help support the genomic data that we're collating. And so our goal right now is to run every, cancer specimen we can access through this to really build our knowledge base of these tissue atlases. And so when we create individual patient atlases, we can then create a biobank or a repository of information for what lung cancer looks like in patients that do well on immunotherapy, and then what lung cancer looks like in patients that don't respond to therapy. And so when we've run hundreds of these samples, what happens over time is we can start to identify patterns in the tissue that predict both response and resistance to therapy. And so we can then potentially in the future identify, the 15 to 20% of patients that will respond to drug, but also in the 70 to 80% of patients that won't respond to these really expensive drugs, what are the alternative therapies that'll help those patients too? So that's ultimately what we want to do, not just identify the responders, but if there is resistance in that tissue and we can see it, we can characterize it, we know it's there, how do we better treat the resistance too? So it's both sides of the coin that we're trying to answer. It is a complex challenge, Nikki. A single slide of a single patient's tissue section, when we've mapped it, is about 10 terabytes of data. And so, to the computational challenges around that are extensive, but we're trying to build a team and the resources and the compute to be able to answer these really tough questions.
Absolutely. I've got a question here about sample sizes. How many samples do you need before you know definitely that this the trend of the biomarkers are indicative of a population size?
Yeah, it's a really good question, Nikki. So, it's almost like this side. We've got a lot of scientists on this call. Sample size is a fundamental question to this approach. The challenge with these approaches is, Nikki, we're throwing about 100 different at the protein level. We're throwing about 100 different proteins onto almost like a paint palette of biomarkers onto every patient's tissue section. Each of these antibodies costs us about $2,000. And so, if you do the maths pretty quickly, it does get pretty expensive pretty quickly to characterize them. samples, I mean, most of the work we do power calculations, and we have an approximate number that we work with, but you know, with growing data sets and building that sample size and the information supporting that, I think we have more confidence in it. Ultimately, you can't what you see at a small sample size is the patient variation. You don't really get the trend data, but we are working with large groups around the world, such as the Human Cell Atlas project, which have profiled thousands of patient samples, and we're providing data into that resource so we can identify not only lung cancer patients in Brisbane, but you know, patients in California and Italy and the UK and Asia. And so because we want to make sure that the patterns we're identifying locally are robust enough to identify patients in other in other geographical locations around the world. So whilst we're developing this work in Brisbane and in Australia, we're working across the East Coast and now with the West Coast too, but we're working with other centers around the world that are developing similar cohorts. And the goal is whatever predictive DNA fingerprint we develop should be applicable in their cohorts too. And so sample size is something that keeps us awake at night.
Another question around the type of cancers you're looking at. So in my introduction, I talked about lung, head, neck, and skin. The question here is, are you looking at those because they have similar biomarkers, or is there another reason why you're specifically looking at those types of cancer at the moment?
These are brilliant questions. So the reason we're looking at head and neck and lung cancer is that's sort of where our major focus is at the moment. But if you look at immunotherapies or the way cancer therapies have evolved over the last 5 years, Nikki, we've moved away from categorizing or giving drugs based on tumor location, right? So an immunotherapy today, the same anti-PD-L1 or PD-1 drug works in melanoma, it works in head and neck, it works in bladder, it works in triple-negative breast cancer. So it works across multiple sites. And so our thinking was, if the drug works across multiple tumor anatomies, then our approach of profiling these tissues should be applicable across multiple tumor types. And so what we're learning in head and neck and lung is helping us inform on skin cancer. The beauty of working across a few tumor types is in lung cancer, we get biopsies of the lungs. And so this is by fine needle aspirate or surgical resection. In lung cancer, we can't get multiple samples over the course of therapy because you're using needles to go into patients' lungs. In skin cancer and melanoma, we can sample that over the course of therapy. And so studies that we're doing in skin cancer today are we're sampling patients' tumors on their skin over the course of therapy. We're getting a punch biopsy, 1 millimeter depth, and we're getting tissue at cure and at progression. And so now we can look at the temporal changes of the microenvironment or the tumor changes over time under the pressures of therapy too. So that can help us inform on, you know, what is evolving in lung cancer, what is evolving in head and neck cancer, where these therapies are the same. And so we're learning from different tumor types. It is hard to move across tumor types, but we're learning from different tumor types, and we're building the evidence base and the sort of foundational knowledge around them. And obviously the lab works in other tumor types as well, but with the sort of major focus at the moment is lung, head and neck, and skin. But we do have smaller projects in ovarian and breast cancer.
Lovely, thank you. Just going to pause there. For those who have joined us recently, I'm Nikki Kinloch, CEO of Cure Cancer. I'm talking to Dr. Arutha Kulasinghe, who's an immunotherapy researcher that's focusing on head, neck, and lung cancers. And we're talking about his spatial biology and spatial mapping. He's based at the University of Queensland, and we've been funding him for first 1 year, a couple of years ago, and now for the next 2 years. If you have any questions, please do drop them in the comments box there. You can direct them to the whole group, or as some of you have been doing, specifically to me as well, and I will weave those into our conversation. Arutha, we have a question here about similarities in the features of lung cancer and in humans and animals, whether there are any. The person has noted this as talked about, sadly, their golden retriever had late stage lung cancer when it was discovered, but no one smoked within their family. So they're just wondering whether this research can then go on to be able to treat animals once you've found the answers for humans as well.
Yeah. That's a really interesting question. So a lot of the immunotherapy trials at the moment, they are being done in canines and in dogs and so on. And so most of our research, Nikki, is almost exclusively in humans, because I think that's where our biggest questions are. But there's a lot of animal work being done at the moment. The preclinical models are all in in animal models, and you sort of need to model the disease in them to be able to understand the impact on humans. And so what we're finding, and there's a lot of really good research coming out of the UK, is that it's a complex process, the development of cancer, but there's a foundational sort of underpinning that's linked with your genetic background, the mutational profiling of the tumor, the inflammation that's associated, and the environmental factors. And so it is a multifactorial sort of situation that results in the development of cancer, and it's hard to sort of pinpoint one sort of individual aspect alone. So it's almost like you need all these individual things to happen together in one environment, and then that sort of progresses into the development of cancer. And so we're learning a lot more about this, especially in smoking, and also environmental factors such as smog and so on, and the impact of sort of chronic inflammation on certain organs. And how chronic inflammation with the right mutational profile could lead to the development of cancer. And so there's a lot of work being done by Charlie Swanton and a few other groups around the world, really seminal foundational studies that we're learning from. Yeah, so I'd say in the canine space, there are immunotherapy trials. I know there are a number of trials in Australia for that. It's hard to sort of, be able to say you develop cancer because of this, because it is a complex disease, and at any one time we've got hundreds of thousands of cancer cells in our blood, but our immune system is generally killing them off, right? And so that's sort of your sort of background around that.
Yeah, lovely. Thank you. In my introduction, I talked about how you are the group leader at the University of Queensland, and you're also the scientific director at the new Queensland Spatial Biology Centre at the Wesley Research Institute. Could you just spare a couple of minutes to talk about what you do from a day-to-day basis in both of those roles?
Yeah. So UQ is my sort of academic position, Nikki, and so that's where I spend 80% of my time. And so we've got a lab there, but over the last period, and obviously with the support of Cure Cancer, what's happened is there's been organic growth in the team of new data scientists and new PhD students that are coming through, really, and the lab's really grown. And so the vision of that was, how can we scale what we're doing? So we're working across different tumor types. And so that's when the Queensland Spatial Biology Centre was established. And the purpose of that was to build the critical infrastructure, the equipment, and the people needed to answer these complex questions. And so a typical day, I'd go to the Fraser Institute and catch up with students and see what's happening in the lab and work with them on their projects, and then come across to the Wesley where you visited a couple of months ago, Nikki, and touch base with the scientists that are in the lab that are generating the data, and really going through a lot of the data and trying to understand the complexities of the data sets that we're working on. And my day starts at around 4 a.m. where we do US calls until about 7, because there's an incredible wealth of information in that space. And then through the day, I'm here physically. I'm either at the University of Queensland or the Wesley. And then at the back end of the day is where I catch up with Europe and some of our Asian colleagues in Singapore and other regions. So it's incredibly collaborative. We work with pathologists, we work with immunologists, we work with virologists, we work with a ton of data scientists. And the data science is this incredible big data problem, right? So you've got tons of data here. that we're trying to mine, that we're trying to ask different questions on. And it's interesting. We sit around, Zoom calls or in person. I try to get everyone in person, once or twice a year, but when we can't, we do Zoom calls. And then we're all speaking English, but we're not really speaking English because everyone's interpreting the data from different lenses. And it's something we sort of fostered after COVID, but we've just kept that going. And it's brilliant when you start to get, an oncologist and a pathologist talking to a bioinformatician. It's like they're speaking different languages about the same problem. And it's just fascinating to sit in that and, pull on different levers and learn from everyone. And so it's an incredible journey to be on, but it's super exciting because we're learning about biology that ultimately links to clinical outcomes. And so, when oncologists say, "Oh, my patient didn't respond to that drug," now we can go find out why. And that's fundamental. And so when we show the oncologists and the pathologists the data, they can, this foreign concept is now visual to them. They can see, in my patient's tumor, that's the area that responded, that area didn't respond. What is it about the biology of that disease that we need to now target? Is there a clinical trial that we can use? Yeah, the days are complex. They often blur, but it's not a job, Nikki. This is a passion for me. It's I think, we need to find better therapies for cancer patients. We need to find more personalized drugs for patients. And so I'm very passionate about this, but so is the team. And so it's incredibly rewarding too.
Yeah, look, absolutely. And I've had the privilege of meeting your team a couple of times now, and I can contest to that. They're all very passionate. Also, what I found interesting in my last visit up to see you was that how clear and obvious you can see the changes in the tumour cells as well. So for those people that may not have seen part of this mapping, it's incredibly scientific, but also beautiful in a way, because you can see a moment of breakthrough that tumour cell is, or part of the cell is being killed. And I know we shared a while ago with Microsoft and Xbox, we had some of their PlayStations wrapped in your work as well. So it's very highly visible, isn't it, when you're looking at it, even from a lay person's perspective?
Yeah, absolutely, Nikki. So I think, growing up, I had a passion about the arts, and so, working across these paint palettes where we're able to, color across these complex tissues is really powerful. It gives us, I think there are messages of hope in there, because we can see, certain immune cells destroying cancer cells, in real time in these tissues. And so there's snapshots of patients' disease, but we can see that the body is trying to fight off these regions. And we've never had the tools to be able to see this before. And so that's the real breakthrough, is that we've now got the tools, we've got the therapies that work in these patients, and we've now got the clinical cohorts coming through where we can ask these really complex questions around, when an oncologist says the drug didn't work, what is it about the biology of the disease? And so we can now pinpoint on the patient's tumor itself potential areas that might have underlying processes that might have driven the resistance to that disease. And so when you think of concepts such as resistance, or it recurred and things like that, those types of questions, now we have visual cues to be able to explain that and dig into the biology behind that. The beauty of the data is that the patterns of resistance that we're seeing they seem to be universal across multiple cancer types. And that makes sense biologically. It shows that there are underlying processes that are driving resistance. And so if we can better characterize them, we can better treat them. And so that's what we're really trying to focus on at the moment. And yeah, I mean, you can see, the other thing we need to sort of think about is these tumor cells are evolving under the pressures of therapies as well. As we do with, microbes, and, if you get a bacterial infection, we give, antibiotics, you kill 98% of the cells, you're left with this 1% that's resistant, and that 1% can grow out. That's the same sort of concept with cancer cells. When you give a drug, you're knocking out most of those cells, but it's those cells that remain that are resistant to the disease that can then spread and evolve and grow because there are no therapies that are effective enough to kill them. And so can we identify those resistant cells early? Can we visualize them? And can we better treat them?
Yeah, absolutely. Thank you for explaining that. We just a reminder, we've got about 15 more minutes left for our lunch and learn with Dr. Arutha Kulasinghe. Any other questions, just drop them in the comments box and I'll include them in our conversation. A question about lung cancer, because sadly, you know, we're seeing a growth of lung cancer, particularly non-smoking lung cancer, and it still is one of the biggest killers in the world. How is your work helping progress the research and then ultimate treatment for lung cancer patients?
Absolutely. It's a great question, Nikki. So right now we're looking at multiple lung cancer cohorts from Australia and the US, and we're trying to access a couple of cohorts in Singapore. And so what we're trying to do is look at about between 500 to 1,000 patients where we've mapped these individual tumors down to individual cells, and then look for patterns within those multi sort of geographic location sample tissues for patterns associated with response to therapy and non-response to therapy. And so if we can do that and develop a panel of biomarkers that defines both sensitivity and resistance, that we can then move those assays forward into the clinic and into the translational space. Because what we can then do is we can say that, look, these 10 biomarkers, when detected on a patient's tumor, identify whether this drug that costs $300,000 per patient per year works or not. And so now all a pathologist potentially needs to do is to stain that tissue section with these 10 biomarkers, and that'll tell them whether this drug works or not. To develop that panel, you know, a 10-plex panel or a 10 biomarker panel would be a $50 test that tells you whether a drug that costs $300,000 works or not. So that's where we want to go, but we need to build that foundational evidence. That'll have, you know, massive implications for, you know, the health spending of multiple, you know, countries that give these drugs. And also, you know, in countries that don't have access to these immunotherapies, it might help tell them about the patient's individual disease in those regions too. And so we're really trying to build that foundational knowledge of that and identify the patterns within those tissues that dictate responses. And so this work is supported by a number of lung cancer advocates around the world. And it's unfortunately, it's one of the, you know, still today, it's got a really poor 5-year survival. So only about 15 to 20% of lung cancer patients are alive today. And a lot of them are alive today because you have targeted therapies for their unique disease. But we know that over time, even those targeted therapies will become ineffective because you're selecting for resistance by giving those drugs. So, you know, how do we stay ahead of the curve and better treat resistant tumors and sort of stay ahead so that we are, you know, you're dying with cancer rather than because of it. And so it's a question that's really, you know, we think about it a lot. And I think one way to do that is you know, getting or detecting lung cancer early, but also then treating it with the best option upfront.
Yeah, absolutely. And we all know Michael Itel, who is a very passionate lung cancer patient advocate. You know, his term is, you call it research, I call it hope. You know, which really strikes a chord, particularly when you are living with lung cancer. Where do you see this going? Because you talked about how you could take hopefully a $50 test to see if this $300,000 drug would work. Once we've developed that and it's gone into countries that have access to immunotherapy, is there an opportunity to go particularly to less developed countries and countries that don't have access to this treatment as well?
Yeah, absolutely. So our goal, Nikki, is to really democratize. So spatial biology is a very expensive tool to develop and characterize tumors, but ultimately we want to democratize the approach globally, because that then, you know, you've got wide adoption of what you can do. what we want to be able to do is to build this knowledge base of information that we're getting from these tests and be able to, so we detect these biomarkers on individual cells, right? And so there's this almost this data cloud of information that sits above a patient's tissue that we're characterizing using that. What the lab's now trying to do is to impute into that slide at a pixel level what that information looks like. And if we can do that, the goal is to then be able to the fundamental stain, or the fundamental stain that's done on all tissue sections, is called an H&E stain. It costs 50 cents, right? Our goal is to be able to read off this really expensive data structure that sits above it that we're characterizing on that H&E stain. An H&E stain costs 50 cents. If we're able to impute that information out at a pixel level on the tissue, that means that a lab in Kenya could scan an H&E is done universally, could scan that, upload the image, and then off the H&E section, this two-colored stain section, we can tell whether these drugs work or not. So that's the sort of pathway, and you know, hopefully over time immunotherapies do get more cost-effective, and as they come off, as other countries start to build up the capacity of these drugs, they will become more accessible.
Yeah, that would be remarkable, absolutely. A question here is asking, well, a statement saying that your focus seems to be identifying whether or not immunotherapy will work. Will these biomarkers also help early diagnosis as well?
Yeah, it's a good question. So our focus is on immunotherapy because we think that's where we're going to get the best benefit for the patients. Currently, we are looking at chemotherapy treated cohorts as well. Early stage is probably where you can make the biggest sort of long-term benefits for these patients. Often lung cancers are detected quite in the advanced stage 3 or stage 4, where patients have limited options. They're often put on palliative care. And so we're sort of starting at the back end where, you know, the survival isn't that great. Can we get these patients that, you know, might have a year or so to live better therapeutic options? I think as we get more information about how cancer evolves over time, we can sort of move this into the earlier stage. We are in head and neck cancer looking at what's called pre-malignant lesions in your mouth. So when you go to your dentist looks at your mouth and identifies areas of inflammation and so on. Pre-malignant disease is sort of what then can develop onto cancer. So you could get stage 1, 2, 3, 4, and so on. Stage 4, sort of your advanced stage. And so what we're doing in head and neck cancer is we're looking at these pre-malignant lesions spatially, and then looking at stage 1, 2, 3, and 4 spatially. And so what that helps us understand is what are the different types of cells that are evolving over this trajectory, and can we prevent, theoretically, those cells from actually coming into contact? And if we can do that, we can potentially slow down the progression of really early stage disease evolving into late stage disease. In breast cancer, for example, you know, you've got mastectomies. We're now starting to look at breast tissue from, you know, 18-year-old, 28-year-old, 38-year-olds, and looking at the evolution of the breast tissue over time. What are the changes happening under the pressure, under the conditions of aging, but what's also happening in the development of breast cancer and the advancement of breast cancer? And so we are doing work in early stage, but we're using that as a lens of what are the changes fundamentally happening in the cells, the structure, the organization, the different communities. And we know that stage 4 disease is very different to stage 1 disease, and it's very different to pre-malignant disease. And so what are those changes? Can we target that? And can we better treat that? So we are doing work in the early stage, but we're sort of working at it from this, you know, this is where we're getting all the tissues at the moment, really late stage, but we're trying to dial that back down into the earlier stage, where if we can identify the changes that are happening at a cellular level really early, then that's potentially therapeutically targetable.
Yeah, lovely. Thank you for answering that. You seem to be making great progress, but in any job, there's challenges. What would you describe as your biggest challenges in your roles?
You got me stumped there, Nikki. I think it's so in Australia, the research that we do here is we run very lean teams. And so really, the teams are, you might have 4 to 5 people in the team, many grad students, but that's about it. That's sort of your structure of your teams. When you look at labs in the US and Europe, you've got armies of postdoctoral candidates and so on. So a lab might be, there might be 60 to 80 people in the lab. So the efficiencies in those labs are a lot better. And so they can scale a lot faster than we can. We do really good research in Australia. And so one way I've had to bridge that gap is to work with those teams in Europe and the US and Asia, where they have orders of magnitude larger resourcing and support. And in the US, if we work with labs at Stanford and Harvard, there's a culture of giving and philanthropy where philanthropy get behind certain cancer moonshots, and they put the capital needed to solve those, or to try to solve those of going that bridge that gap without physically moving to the US is to work really efficiently with those teams. And so that's why my day starts at 4 a.m., Nikki, to work with those teams, so that we can learn from the best and work with the best, but also they want to know what we're working on, we want to know what they're working on, but also the challenges we have can be we can find solutions to those by working in that sort of international collaboration space. And so that's one of the challenges that we have, but I think we found a solution that works for us, and also to live in one of the best countries in the world.
Absolutely. And you talk about team science and interdisciplinary teams, and I think we've touched on it, but obviously collaboration is incredibly important in the work
Absolutely,
that you were doing.
Nikki. I mean, when we sit around a table, even at a lab meeting, you've got PhD students, you've got data scientists, bioinformaticians, virologists, immunologists, medical oncologists, all around. We cannot cure cancer individually. It needs to be a team science approach. We've learned that during COVID. And so, it's sort of this flat management style. Everyone has a voice. We all learn from each other. And it just works, Nikki. It's just, when you work in a collaborative space where our goal is, and I think that's shaped by the advocates we work with. So Michael Eitel, for example, whenever he goes for a checkup, he comes into the lab, he talks to everyone, he comes into lab meetings, hey, what are you guys working on? What are you spinning down those cells? Or, he's got all these, crazy and wacky ideas, but it's great because we have patients at the center of our research. And so everything is shaped by them, and it's because of them. They're our why when we wake up in the morning. Simon Sinek speaks about, defining your why, and our why has always been patients and understanding and better treating them. It's something I learned in the US, where if you have patients at the center of your research, everything else just, motivates itself. And so it's an incredible journey because we're all learning from each other. We're all moving together. And I think team science is the future of biomedical research. You need multidisciplinary and interdisciplinary teams working together. Some of these problems are maths problems. Some of these problems are immunology problems. Some of these problems are oncology problems. Each one of us cannot answer it individually, but together we can, and we can try. So I think that's where we're at the moment.
Yeah, lovely, thank you. And we have several of our donors on the call today. And obviously funding is always an issue, but do you have any words for them? Because I know many of them have supported you over the years, and particularly excited to see how you and your team is developing in spatial biology research.
Absolutely, Nikki. We're incredibly grateful, Nikki, for all the support. I don't just say that, I mean that. And I think our lab, at the end of the day, early career research, that we have all the crazy, nutty ideas, but we can never get the funding to make those, actually move those ideas forward. Often incremental gain research is what is funded, and Cure Cancer funds blue sky type of research. And I can put my hand up and say the reason my lab is as it is today is because of the funding from all the donors that support Cure Cancer and everyone behind the scenes that help to support our research. We have all the ideas as early career researchers. We have all the, it's all buzzing around in our head, but we can never get the funds to get them off the ground because we don't have the track record to be able to go for national funding. And so without funding early career researchers, a lot of those ideas die, you know, on the bench. And so, you know, I've got a lot of colleagues that have left science because of this. And I, you know, every researcher that you fund, you know, can potentially make the breakthrough that we need to, you know, better treat these patients. And when we get the samples from the hospital, they're often warm from, you know, bloods or tissues. And we often tell our grad students, you know, it's like liquid gold that we work with. And so everyone's, you know, incredibly motivated around this work. And you know, it goes back to the, you know, the funding that's enabled by Cure Cancer.
Look, thank you. And thank you to you and your team. For those that aren't aware, probably about a month ago now, The Australian also did an editorial on Arutha and his team and the incredible work they're doing. It is groundbreaking. It is innovation. It's exactly what we fund, and we're incredibly proud of you and of all of you have achieved. You know, how wonderful would it be if we could democratize cancer, and also if we could die with cancer rather than die because of it. So on behalf of everyone here, thank you, Arutha, and thank you to your teams for all of their hard work over the last 5 to 10 years.
Thank you, Nikki.
So that's the end of our session today. I hope you have enjoyed it. As I said earlier, we will share this recording next week. If you do have any other questions, please drop them to info@curecancer.com.au. Thank you to those that have generously supported our research and helped fund research like Arutha's. As you can see, it's incredible research and it is changing the way we treat cancers. Please join me at the same time on the 12th of September for our Lunch and Learn with Dr. Jen Karantai, where we'll be talking about their work on liver cancer. So until then, have a good afternoon and stay safe. Thank you.